And I now would like to request Dr. Anjani Devi, Vignan's Foundation for Science and Technology Research, to introduce the next speaker. Ma'am, please.
Good morning, everyone. I take immense pleasure in introducing Dr. Prashanth Suravajhala. Dr. Prashanth Suravajhala is Dr. N. Prashanth Suravajhala is Senior Scientist at Birla Institute of Scientific Research, Jaipur. He obtained his PhD degree in systems biology. He went on to gain more than 7 years of postdoctoral experience across 4 different laboratories. He has interests exploring the known unknown regions in human genome, primarily working on the top-down systems biology approaches of hypothetical proteins. For the last 5 years, he has developed an interest in long, non-coding RNAs in humans, elucidating the mechanisms underpinning small molecular interactions through clinical exomes. He founded bioclues.org in 2005, India's largest bioinformatics society, which advocates open access. The organization aims to bridge mentor-mentee relationships and has been successful in mentoring as many as 2,000 graduate students across 30 countries. He is also associated with bioinformatics.org and a host of other humanitarian organizations like Rotary Community Corps. Now, I request Dr. Prashanth to deliver his talk on Systems Genomics of Prostate Cancer, Indian Perspective. Sir?
Thank you so much, Dr. Anjani, for the kind introduction. Is my voice audible, please?
Yes.
And screen visible? Yeah. My internet connection is a bit unstable today. I don't know why, but I apologize for this. I hope it is audible. Yeah.
Yes, sir. It is audible, sir, and your slides are also visible.
Yeah. Thank you. Yeah. So at the outset, I would like to congratulate and thank InSyB organizers, especially Professor Kavikishore sir, Doctors Rao, Anil, Jalaja, Abhinav, and Tarun Pal for graciously inviting me for this particular conference. InSyB has been a wonderful conference series, especially this one. It's been about a conference with over 600 participants, which is quite amazing. I feel greatly honored, to deliver this particular talk after GPS Raghava, who is the doyen of bioinformatics in India and also the past president of BioClues. Thank you very much, sir, for this wonderful talk. So my particular talk probably, would delve on the history and rationale behind the prostate cancer project. So where are we now? I'll just, try to summarize the things. And I'll also talk about the kind of problems and challenges that we are probably looking into these directions, and the whole exome sequencing spectrum of prostate cancer, which we have started in relevance to India, and the kind of inherent pathways, and the big data challenge and the machine learning heuristics that we are just trying to attempt. We have just started attempting it. But into COVID times, we couldn't finish it. And then I'll also talk about the long non-coding RNAs and the regulatory elements associated with this. So I come from Birla Institute of Scientific Research, which is a sister concern of Birla Institute of Technology based in Ranchi in Jharkhand. Our current chairman is C.K. Birla, who is at the bottommost photograph here. And ours is a philanthropic institute, just like Tata's and Birla's. And we have got a state-of-the-art facility based in Jaipur with 2 departments. One is biotechnology and bioinformatics, and the other one is the remote sensing. We also have got a planetarium, state-of-the-art museum, and a big auditorium. And our current director is Dr. Punendra Ghosh, and Dr. Krishnamon is our head R&D. who is also my mentor. We're a group of 14 scientists, 7 PhD fellows and 2 postdocs, of which 6 PhD fellows are working with me. So before I set up this particular thing, so I just try to tell why this prostate cancer project came into came into our mind, came into the objective hypothesis that we have set. So it was way back in 2013 and 14, during my third postdoc heydays. I've had an illustrious career of about 7.5 8 years of postdoc, and I've worked across 4 different laboratories. So during my third postdoc in 2013, I just returned again back to Boston and Boston to Denmark with a brief stint in India during that time. And my postdoc mentor, Haja Kadamidin's lab, they were working on something called feeding deficiency traits. So they would probably overfeed the mice and overfeed the eutherians, like cattle, buffalo, and all those animals sitting in the animal genetics labs, and they would try to look what are those particular trait specific genes that are probably associated with those particular animals. So like as you could see here, there's a mice, this was the earlier photograph. It is overfed and it becomes obese. And likewise, the cattle as well. If you just keep if you keep raising cattle, it will keep on eating and eating and eating, and the farmers would probably think that it would probably eat more milk. But one thing happened when these animals are really overfed. The 2 PhD fellows in that lab, when I was doing the postdoc, their job is to feed it, collect the serum, from the from those animals, and try to see the differential expression count in those particular animals, and see if there are any genes that are upregulated or downregulated. So in nativity, if you really see, if we really eat normal, we assume that we are healthy. If we eat less, we are unhealthy. And if we eat optimal across different time periods, giving some breaks, again, we are healthy. But if we are eating too much, the unfortunate thing is we become unhealthy. There are a lot of unnecessary metabolites prevailing in your body that might make you obese. This we know, but we didn't know whether or not those animals would become prostate. Those would be affected with prostate cancer, and that was how this particular project started. And I thought if there are any obesity related genes that are associated with prostate cancer. And then after I had another postdoc between 2014 and 16, and then I just sort of coming back to India, and that was how I started writing a grant. And this grant is a major grant of 1 crore, which I've gotten through Novartis and BrainPan, that I'm associated with it. So this particular prostate cancer, we just asked if there are any mutations relevant to India. That's because 1 in 9 men are diagnosed with prostate cancer during their lifetime. It's usually the instance is above remarkable, and with an age factor precedent set above 55 years. And it is the second leading cause of death globally, and it has become third most prevalent cancer after breast and oral cancer in India. And it's rapidly growing. There are different things. I mean, the life expectancy is going to be declining, and there has been a declining fertility rates as well. Although at this point of time we are only looking into the so-called somatic mutations, not the germline mutations, because we haven't really understood what are the mutations specific to prostate cancer in Indian subpopulation. So this is how it is. The take-home message here is, as for the Globocon statistics for 2018.
Sir, your voice is not audible. Could you please unmute yourself, sir?
Yeah, can you hear me now? Yeah.
Yes, sir, you're audible now.
Yeah, someone
Please go
muted me. I'm really sorry. So can you see the screen as well? Yeah.
Yes, sir. Yes, sir. The screen is visible.
So and again, this prostate cancer is, quite seen in African Americans. It's because, it comes with the diet factors as well, because probably they eat a lot of red meat. Whereas Asian Indians, many Asian Indians are comparatively vegetarians, if not vegetarians, or probably at least, people eat, fish when compared to the red meat. So we Asian Indians eat a lot of, broccoli kind of things. So we are not, that inherent to, prostate cancer. But if you really see the far west, where, there's a lot of, meat eating people, especially in the Americas, so prostate cancer is absolutely visible over there. So, so if you really look into the risk factors, so there's always an association between fat consumptions, and they could be also be associated with countries with greater per capita milk consumption. Okay, so the most important thing here is there is no association between smoking and alcohol. I mean, it is assumed that if people smoke a lot, maybe people drink a lot, they might be associated with cancers like prostate. But just imagine a person who is getting a prostate risk at the age of 55, and maybe you would have really started drinking right from his age 21. He would not have really gotten just cancer just because he's over drinking or he's over smoking. So that assumption is absolutely a kind of a false entity. And of course, low per capita means countries with low per capita has got lowest risk of prostate cancer, just like in India, or probably some of the African countries. But there's also an assumption that in Americas or Australia or UK, they have got higher risk of prostate cancer because they've got high per capita income. And if you really see the detection things, detection is usually done using prostate serum antigen and distal rectal examination. There's also something like biopsies. There are also some markers that are usually done in the pathological lab, like the kallikrein HK3 markers, which is a human kallikrein gene. And there are also tumor markers. There are also some tumor necrosis metastasis factors, which is what we call it as a TNM grading for any cancers. But here for prostate cancer, there's something called Gleason score classification. So if a Gleason score is above 5, then that indicates that the prostate cancer risk is higher. And below 5 is nothing but they're all the benign tumors. And there are some urinary biomarkers. One is a well-known long-known coding RNA called MELET1, which is well studied. So what we have done, when we came here was we have attempted to write this particular grant to DBT, and unfortunately it was not true. But I thought, since I was associated with BrainPan, we tried to contact Novartis and other companies. And that is how we got this major grant. So we started with a pilot of 10 samples that was escalated to another 10 samples, about 20 samples, and another 110 samples are on the anvil, the sequence. So what we did was a whole exome sequencing of these 10 samples with about 5 benign cases and 2 with intermediate Gleason scores and 3 Gleason scores about 7. And the ethical clearance was judiciously taken from the Birla hospitals, thanks to our clinical collaborators. And these are the cases. We have taken an inclusion criteria of age about 55, of course, with a Gleason score of 6, with a prostate serum antigen of 4 nanogram per ml. And these are all the malignant. There's something called benign prostatic hyperplasia, where the control samples have got Gleason score less than 6, as I told you. And if you really see, we have developed a lot of, good number of, bash-based pipelines to benchmark these whole exome sequencing pipelines. So you simply give a FASTQ file, and then at the end of the day, you'll get a lot of variants with SNPs that are associated with different disease conditions. So we have benchmarked this pipeline, which is MENA et al. 2018, which is highly downloaded protocol, which was widely used by more than 25,000 users globally. And this is how the classification is. We got a good number of reads. with a good size across, and all of them were quality checked. And if you really look back into the basic nitty-gritties of these particular samples, out of these samples, all samples are all patients are all vegetarians actually, and yet we were a bit surprised, how come many of these particular samples, although being vegetarian, they have got the prostate cancer risk. So if you really talk about the results, we asked that there are some high impact variants across high grade prostate cancer tumors that are specifically inherent to Indian subpopulation. So if you really see, we found about 30 causal genes that are responsible for prostate cancer in India. So some of the well-known, BRCA1 and BRCA2, which has got, which are also associated with breast cancer, that are also seen in men, as you could see here. There is also a CTNS gene, which is associated with vitamin D factor. Although we receive a lot of sunshine, even many of these prostate cancer patients have got this CTNS mutations sitting over here, and also some PUSH2A and XP mutations. These are the DNA mismatch repair proteins. There are also some proteins like GMP-TAB, which are involved with transcription factors as well. And of course, the TP53, which is a tumor suppressor, which is also associated with obesity. The COL6A1, which is primarily a copy number variant, where that particular chunks are also seen across different genes. That is also associated with obesity as well. And we have also found some of the other genes that are associated with mitochondrial function as well. So OPA1, which is a well-known mitochondrial gene as well. So which means it's also probably inherent, because there could be mitochondrial mutations that might be transcended from the mother as well. So OPA1 is one of those particular victims. So as you could see, the square boxes are all those genes that are seen in Caucasian or West, and the ones that are in circle, these are all those mutations that are primarily seen in Indian conditions. So this shows that in India, diabetes is also associated with prostate cancer risk. And this is what we have inherently done. We have checked across a union of intersection of variance between cases and controls. And we have done some proved statistics. So these are our first authors, Ayam Gupta and Nidhi Shukla, my PhD fellows, who have done this particular work amazingly, and which we recently published this. So the part one of the conclusions is our patients in our cohort are vegetarians, and they are yet prostate victims. Our pilot study revealed that there are mutations associated with obesity, diet, mitochondrial dysfunction, and as well as vitamin D deficiency. There are some tumor suppressors. There's also a copy number variant like COL6A1 detected. You might ask, in a whole exome sequencing pipeline, usually you cannot identify copy number variants, because copy number variants are, bigger chunks of mutations, anywhere between 40 bases to up to 1500 bases long, and they're all those segments, that are seen across, different genes. So usually a whole exome sequencing cannot detect it. But we have tweaked our pipeline to identify some of these copy number variants. Some of the copy number variants are seen. And there are some diet-induced obesity genes, like these three, and also CTNS gene. And we have come to a conclusion that, yes, we have developed a conceptualized framework to prioritize certain mutations for prostate cancer risk in Indian subpopulation. So that project was, that was the part one. And I've been, somehow interested in long non-coding RNAs because, my PhD, I formulated something on the hypothetical proteins. Thanks to my wonderful mentors, we have developed, some databases and some platforms to identify whether or not a hypothetical protein, can be, classified in a good way or not. But eventually, many of these hypothetical proteins have turned themselves to pseudogenes, or some of them were misannotated as long non-coding RNAs. Okay, and that is how I tried to ask if there are any known unknown regions in the genome, that can be essentially tagged as long non-coding RNAs, and maybe who knows, long non-coding RNAs can also be associated with prostate cancer. So This is what we try to walk through the genome story. So if you have any gene somewhere here which is associated with say your diseased phenotype, so in this case a prostate cancer. So this is an ensemble genome browser where this entire region is a gene responsible for prostate cancer with a contig like this. As you could see here, there are some yellow blocks, which are in fact enhancers, as you could see here. Okay, and there are some blue ones which are the CTCF motifs. Okay, and there are some squared boxes somewhere over here which are the long non-coding RNAs, as you could see here, these ones. Okay, so which means if you have a hypothetical, if you have a gene of interest for any diseased phenotype, if you walk through the genome, usually the 5' untranslated region up to 5,000 region or 5,000 bases of upstream of that particular gene, you will find these regulatory elements which might influence the way that particular gene works. So they might ensure that just because there is a regulatory element sitting over here, this regulatory element might have an influence on how that particular gene might harbor certain mutations sitting in it. Okay, so this is what we call it as a genome walking. So this is what a BRCA2 gene, and incidentally, Vignan University students are also doing this particular genome walking story along with Nidhi Shukla. So this is what we are trying to work. And a visiting graduate student, Narmada, she has performed this particular genome walking story as well. And we have identified some long non-coding RNAs and long intergenic non-coding RNAs as well. This is still ongoing. We have just characterized some of these long non-coding RNAs, which we are still trying to attempt and come to a consensus and ask if there is any long non-coding RNA, that could also be associated with these diseases. So this is a long non-coding RNA, non-HSAT 053810. But eventually, you might even ask, Prash, why is it you're always talking about long non-coding RNAs? So there could always be some other small non-coding RNAs like the pvRNAs or the microRNAs as well. So microRNAs, although they're pretty short, they would fall short of mutations because they're highly conserved. They're anywhere between 18 to 24 bases. So being 18 to 24 bases, the chance of having mutations in them is very less. So we try to ask if there are long non-coding RNAs, and eventually there are also some pvRNAs. pvRNAs are anywhere between 24 bases to 45 bases in length. So we have also found this pvRNA 3359922 as well as of now. So we are just still working on that. Nidhi and I they are working on this. So and something exciting popped up in between. So this COVID came and all our lab activities have really stopped. And then I started working on this SARS-CoV-2 along with Keshav Singh. So what happened was when this COVID came into picture, we again asked if there are any receptors sitting somewhere in the host genome where in addition to the ACE2 and TMP-RSS2, which are the receptors in the host that invite SARS-CoV-2 to enter into the host, are there any long non-coding RNAs that are probably sitting over here? Are there any transpressed, or are they really sitting over in the host that might be associated with this SARS-CoV-2 as well? And then we instantly, from the data that we have observed, that we have found that this non-HSAT 23988 is highly expressed in prostate as well. And this long non-coding RNA is in pursuit of this particular receptor-mediated entries. There are also other long non-coding RNAs that are substantially expressed. The reason why I'm talking about this long non-coding RNAs is because they just sit in the host, they invite SARS-CoV-2, and the long non-coding RNAs with the help of ACE2 and TMP-RSS2 try to receive it, and as soon as they receive it, the hijacking process of SARS-CoV-2 comes into shape. So it tries to eat away all the mitochondria, and that's one of the reasons why people will not be able to breathe properly, and that results in cellular death and finally asphyxiation, and finally the person dies. But in milieu, there is there is most importantly in a lot of prostate cancer associated long non-coding RNAs in this particular process. So the part 2 of this particular story is there are some pVRNAs also in addition to the long non-coding RNAs. We try to identify whether or not these long non-coding RNAs have an influence on this particular gene, whether or not they interact with the proteins. And we are just still trying to identify if any of these long non-coding RNAs or pVRNAs could really make up a good story here, where they could be associated as interaction partners. Thereby we could develop a kind of a viable prognostic marker towards development of prostate cancer risk in Indian subpopulation. And recently, what we have developed is, apart from this hijacking story, we have also come up with a new hypothesis called transcription hypothesis. This manuscript is communicated. We are hopeful to get an answer sometime by this year end. So there is a non-structural protein 15, which is over here, NSP15. So what happens is this non-structural protein 15 has got a highest chance of transgressing the long non-coding RNAs, because if you simply blast this NSP15, it shows resemblance with a lot of non-HSAT 255167. And what happens is, as you could see, these are those particular bases that are absolutely transgressed. When I say transgressed, this long non-coding RNA is sitting in the host. NSP15 takes control of the virus and the host. And in the milieu, the long non-coding RNA bases are eaten away by these viral particles. And that is how this CTNND1, which is a gene called connectin, catenin. This particular catenin gene is also known to be associated with this long non-coding RNA. And this is how the transcription hypothesis unfolds. So, what happens is there are also other receptors in addition to the transmembrane protein RSS1 and ACE2, angiotensin converting enzyme. Is other receptor like interleukin 17, which is sitting over there, where these long non-coding RNAs are being controlled by these particular receptors. Okay. So what now? This is my final part of the story. So now we have annotated a lot of genes. We have identified some mutations across 30 genes, out of which about 7 genes are primarily responsible. A couple of them are associated with diabetes. A few of them are associated with mitochondria and all. So can we come up with something like a predictive analytics? Can we come up and say that yes, we have identified some genes responsible for prostate cancer risk. Now, what if we have some population that is coming up in the near future? Can we say that yes, these are the mutations that might be associated in this given subpopulation if we have a patient coming with such disease phenotypes. He might be obese, he might be prostate cancer risk, he might be associated with a mitochondrial dysfunction, and if he has a scrotal pain, can we just by looking at his vitals, can we really say that yes, he might harbor these particular mutations? So we're trying to look into the precision scale. That is what we are trying to ask. And so we have a whole exome sequencing data. We have a list of variants over here. And in part 2, we have got all the regulatory elements, the long non-coding RNAs, and also the RNA-seq, the whole transcriptome sequencing data, which we are currently doing along with. And now the part 3 is a deep variant approach, which we have attempted it. Our other program is still running, which we are just trying to ask if we can combine this and come up with some predictive analytics. You can also see the screen, right? Yeah.
Yes, sir.
Yeah. So so this is what we try to ask, and then there are no diagnostic methods that are available in the wet lab today. Even if they're available, the diagnostics would be very much costly. It's not a kind of an easy thing treating a kind of a prostate cancer patient. It involves a lot of monies as well. So as you could see here, the costs are absolutely very heavy here. Even in India as well, right from biopsy till getting the digital rectal examination done, it takes anywhere between 20,000 to 35,000 rupees, which is pretty expensive. So since this is a largely funded project, coming from BrainPan and Novartis, so we thought of coming up with a kind of a kit. Can we develop a kind of a simple pregnancy like kit where because many men are usually stigmatic, they don't usually come and open up and say that they've got some pain in scrotum or they've got some pain in rectum. So can we come up with a high turnaround time kind of kit where with the simplest urine they could probably check whether or not they are at prostate cancer risk. So although MALAT1 is well-known urinary biomarker, it is not associated with prostate cancer. It is associated with genitourinary cancers. So we are just not trying to attempt. We are just not trying to ask if we can really come up with a new cancer-based prognostic kit. It can even be a kit. It can even be a kind of an NGS panel, which is pretty cheap. So simply taking up and checking from those particular measures whether or not those particular genes, if they're present in blood samples, then we can say that the person is prostate cancer risk. So so the final lessons and final summary over here is we have identified about 10 long non-coding RNAs and 1 pbRNA. Although we have found about 10 long non-coding RNAs, we have jotted down to only 3 at this point of time. Our analysis is further leading to understanding the regulatory elements, the CTCF motifs and the promoter flanking regions, open chromatin elements, the enhancers and the transcription factor binding sites. Our present results, probably support the multi-gene panel testing because we might have many genes where we might take the coding genes, the non-coding genes that are associated with diseased phenotypes like prostate, genitourinary cancers or whatever, and come up with a kind of a panel. But we need some kind of a deep variant approach. We need some machine learning approach which we are trying to work on. So that would probably set a precedent to work on the immediate goal, like taking the deep variant based approach, taking the Gleason score attributes, and the distant goal would be to look into the hereditary conditions. Say, for example, in India, if a person aged 55 is prostate cancer risk, what is the percent chance that his 26-year-old son might also be associated with prostate cancer, okay, when he turns 52? So that kind of things we are trying to ask, and we are trying to simply work towards these particular conditions. And we also would like to look into the role of factors like diabetes, obesity, vitamin D, and other dietary elements. Where there are some genetic factors that are associated as of now. There are some Indian specific variants as well. There are some genetic changes that are responsible for this prostate cancer as well, like the CNVs and the PON mutations. Of course, we're also looking into the long non-coding RNAs like MALAT1, taking into this thing as a picture. And we are also trying to ask some questions. What if a benign prostatic hyperplasia patient who has never been considered as a prostate cancer victim, what is the percent chance that he might be prostate cancer risk in the future? And what happens if a person who is with hemorrhoids, who is down with fistula, who is down with piles, what is the percent chance that he might also be prostate cancer risk by the time he turns 55? Okay, so these are all the predictive analytics that we are still working on. Unfortunately, in India, we are still in infancy at this point of time. So I think I'll probably end here. I'll be happy to take some questions here. But before I end, I would like to thank Kaviteshwar sir, Dr. Srangarao, Anil, Jalaja, Abhinav, and Tarun for inviting me, and also Asif as well, who has been graciously helping BioCruise as well. This work would not have been done without these two wonderful people. Nidhi, who is a women scientist fellow and also a PhD fellow. I am Gupta as well. Mamata and Narmada, they were graduate students who worked, and now Bumandeep, who is my another PhD fellow, where Sugunakar is the main supervisor. She's starting working on prostate cancer. And these are the other PhD fellows who are working on different projects like vitamin K and the known unknown regions. Sanchita is also here, and Shalini working on the vitamin K. These are all the clinicians. In fact, clinicians' role is very, very crucial in India because without them, we would not have gotten the samples. My full credits to Devendra Sharma and Manish Vijay, the clinicians, urologists and pathologists working over there. Amrit and Saurabh, through which we got the brain pan funding through Novartis of 1 crore. And of course, Ashwin, who is my co-PI. And Krishnamohan sir is my mentor. Obal, my collaborator, and Dr. Praveen Mathur, who is a well-known pediatric urologist. And of course, Kavikishore Sarangiri sir, who's whom we cannot really forget. They have been absolutely wonderful people to bank with. And Vikram Singh Chauhan was the one who ensured our ethics clearance were in place, and Jyotsna Patra as a part of a practical consortium, and Nirmal Lohia sir, who has been a wonderful person. And Subbu, my collaborator, of course, and Vinod Nigam, my collaborator. So thank you so much. The most important thing, the bottom line, I'll just take just 30 seconds here. There are about several people attending here. I know, we have really faced a Herculean task of 2020 this year. I request all of you to learn to collaborate, not to compete. This is the time, where we all, come together, join hands, join forces, come together and say that, we are Team India. We are all, working towards, for development of science. If we start thinking that, the other person is, competing with each other, probably we should also compete, then that's absolutely a wrong thing. And that is where, these 3 C's, come into picture. Always, try to collaborate, try to converge, try to learn from each other's, expertise. And that would really bring out, a wonderful, consistency. And that is where, we can probably, combat, COVID-like, viruses in the community. Thank you so much. Thank you.
Thank you, Dr. Prashanth. Now, the session is open for discussion. Participants.
I have a question. This is Kavikishore.
Yeah, please, sir. Yeah, thank you, sir.
In 110 samples that you have taken for prostate cancer analysis,
yes, sir.
You said, most of them are vegetarians, but still they are getting, the prostate cancer or at the risk of prostate cancer, How many samples you have taken from the non-vegetarian group?
Sir, that is where, in fact, our study is a very pilot study. So this sample is only from 20 samples. The study is only from 20 samples so far. The other exome sequencing is still being done for about other 20 samples. So total about 40 samples we are going to do now. Out of 20 samples for exome sequencing that was done, 19 samples were vegetarians from our study, from our understanding, when we counsel them. And one other person is a non-vegetarian, that is from our understanding. So this is a kind of a very beginning to be understood, sir. It's only, a very crude, thoughts, going through our minds. What would happen, if there are non-vegetarians that are really obese, and what would be their fate, if they are really a prostate cancer risk? So, yeah.
The long non-coding RNAs and pvRNAs that you have identified with prostate cancer, these non-coding RNAs and pvRNA have also been identified in other countries, in other groups?
No, sir.
Only in India?
No, yeah, it's only with respect to Indian subpopulation. So, in fact, we have seen these long non-coding RNAs only relative to Indian subpopulation, but one long non-coding RNA, which is 2399AA, that is from BRCA2 actually, that is present somewhere in the upstream of this BRCA gene. That is quite common, and that is rigorously seen across other patients as well. But sir, this is a kind of a walking story, which is a genome walking story, which we have identified from a whole exome sequencing. So we are not sure yet whether or not this long non-coding RNA is differentially expressed from the whole transcriptome shotgun sequencing where our study is underway. So we have given these particular samples to our service provider. So once we get those RNA-seq data, it would be wonderful to see if those long non-coding RNAs are differentially expressed in those genes. So that is where we are trying to begin, trying to understand.
Yeah, and also I have a foolish question to ask.
Yeah, please, sir.
Whether it would be possible for us, to use some of these long non-coding RNA markers, or the pRNA for predicting, the prostate cancer. Can we use it as a diagnostic tool to predict, in advance, ahead of times, to the prostate cancer?
Right. I mean, this is a wonderful point. So it's not at all a foolish question. That's where we are trying to understand. So there's something called human kallikrein markers, which there's a gene called HK3 that is well known for prostate cancer or rectal risk. So what we are trying to bridge the gap here is, let's assume that we have identified a long non-coding RNA that is absolutely responsible for prostate cancer. Now we are going to ask whether or not this long non-coding RNA is circulating in the blood as well, or are they really associated to be considered as circulatory biomarkers? That is number one. And if they are circulating, then there is a chance that these circulating biomarkers can also be associated with the pathologic manifestation like the human kallikrein gene. So then we would try to ask if any of these long non-coding RNAs have an influence on some kallikrein genes like HK3, where they can be subjected to development of some prognostic markers. So this is a very the study is absolutely in a kind of an infancy, sir. We might get some exciting results sometime by the end of next year. Because we also have a very good plan to come up with a development of a good kit to Novartis and BrainPan, where the study is underway. Because it's largely funded, and the current problem that we are trying to face is the lack of samples. Unfortunately, we are not really getting samples as of now. But your question is absolutely very valid, sir. Thank you for asking.
Yeah, thank you very much, Dr. Prashanth. I have thoroughly enjoyed it.
Hello, Prashanth. One question from Professor Krupanidhi.
Oh yeah, please, sir. Please, sir. Please.
Yeah. So Prashanth, I've been following your talk. It is quite interesting, and I wanted to get to know, because you mentioned 10 long non-coding RNAs. So they're, as I presume, I may be wrong. So they are only prognostic markers, not causative features, no?
Yes, sir. I mean, as I told you, these are all those long non-coding RNAs which we presume that they have identified or they are associated with the genes responsible for the whole exome sequencing study. Yeah. So in order to make it further, we need to validate their expression. We need to validate their regulatory role and the differential expression from the RNA-seq data as well. Mm. So the RNA-seq data for about 6 samples is underway right now. And we might do RNA-seq for another 20 samples sometime during the next few months, sir. So once we identify these long non-coding RNAs from these differential expressed count as well, then we can say that yes, out of these 10 long non-coding RNAs, maybe 2 or 3 or a handful of them could be associated with prognosis. Yeah.
So that can be linked from the childhood onwards.
No, these are all the somatics, sir. I would say. I mean, unfortunately, we cannot say, because, and of course, I mean, if we really say, that might really come up with childhood. That would, probably be a question of yes or no. That's because we don't know whether or not any of these particular mutations are seen in these long non-coding RNAs as well. That we need to identify only from RNA-seq. Although genetic variation is seen in the long non-coding RNAs, the risk of attributing the mutation specific to long non-coding RNAs is very minimal from the whole exome sequencing study. Yeah. Because from the whole exome sequencing, we'll not be able to identify the long non-coding RNAs at all. So it's only
these long non-coding RNAs will be having influence on the causative genes.
Yeah, very much. That's where these long non-coding RNA protein coding gene interaction studies are underway, sir. So so that's what we have tried to attempt. Yeah.
Is there any transcriptomics coming out of non-coding RNAs? That is non-coding gene, that SYN and line.
Yes, sir. That is where our studies are underway. Our samples are being analyzed now for RNA-seq, the transcriptome study. So we are looking forward to see their expression in these particular samples as well.
Yeah, okay, thank you. There's a lot to know through you. Thank you.
Yeah, thank you, sir. Yeah, thank you.
Sir, any more queries from the participants? Okay, if not, thank you, Dr. Prashanth. You have given us a clear-cut idea on the prostate cancer, reasons for prostate cancers. Besides, you have given us the idea about what are the risk factors associated with the prostate cancer. Thank you, sir. Thank you for your enlightening speech.